Hello everyone and welcome to another webinar from Diagnostic Solutions Laboratory. Today's webinar, Understanding C. difficile, New Clinical Insights from Recent Research, is presented by Dr. Tom Fabian. Dr. Fabian is a clinical laboratory consultant, translational science expert, functional nutrition practitioner, educator, and speaker. He is a former biomedical research scientist with deep expertise in the role of the human microbiome in health, chronic disease, and aging. As a leading expert in translational applications of microbiome research in functional medicine and integrative health settings, Tom's primary focus is on providing educational resources and consulting services for practitioners and scientific advisory and consulting services for clinical testing laboratories. At the end of the presentation, if we have time, Dr. Fabian will answer some questions, and any questions not answered will be sent to our clinical team after the webinar. Whenever you are ready, Dr. Fabian, you can start.
Great. All right, well, thanks so much, Ali, for that introduction. And I also would like to thank everyone for joining us today. So I know we get a lot of questions about C. diff. It's sort of one of those enigmatic organisms. And clinicians often don't know exactly what to make of it, how to interpret it, et cetera. So we're definitely going to dive into that today, focusing on a particular clinical case, as well as a lot of the newer research that's been coming out, which really helps to provide us a lot of context around C. diff and overall helps to contribute to better understanding of C. diff, and especially in terms of providing better clinical insights for individual patients, and also hopefully better outcomes as well once we take that information into account. So I'm going to go ahead and dive in here with the first slide. We're actually just going to dive in right away to start familiarizing you with the particular case example that I'm going to keep referring to throughout this webinar, because it's a great illustration of what we commonly see with C. diff in terms of the patterns on GA map and also the related scenario for this particular patient. So this particular case report was for a 40-year-old female who had a significant history of food sensitivities, as well as other food-related symptoms that seem to appear within the period after eating, after meals, and that would include significant headaches, pretty common headaches, skin rashes, flushing, and even some neurological symptoms occasionally. This patient had also complained of chronic constipation and also significant fatigue. So fairly common symptoms that we often see in clinical practice. Just a side note is this patient had a separate test run through a different clinician initially related to these symptoms, and it was a LabCorp test for C. diff. So we'll kind of get into some of the differences here, but that test looks at a bit of a different aspect of C. diff, testing for the production of the toxin. And in this case, that test was negative. But as you can see on the next slide here, when the GI map was run, both toxin A and toxin B genes were detected for this particular patient. And both, of course, at a high level, as you can see here. So the question is, in the absence of sort of the classic symptoms, what may be going on here? What's the overall interpretation based on the levels of C. diff, based on other things that we can see on the test, and how that may relate to this patient's symptoms? I'm just going to go through the rest of the report here. You can see the parasitic pathogens, viral pathogens were negative. H. pylori in this case was also negative. If we look at the normal bacteria flora section, we see a few imbalances going on here that may be pretty significant for this particular patient. We'll go through some background information that will help you understand what's going on in this section and how that may relate. We see one of the markers, Enterobacter, is elevated. And also two of our markers in this section, one for Akkermansia mucinifila, is low at less than detection limit, less than DL. Then also Faecalibacterium prausnitzii, in this case, was quite low. And as we'll talk about, that's quite an important species. Something is often seen as being low in C. diff infections. So there may be some connections here in terms of what we're seeing in the normal bacteria section. Looking at the opportunistic bacteria section, we can see overall pretty significant overgrowth pattern with high bacillus, high enterococcus. Both of the Pseudomonas markers were elevated, high staph, and also high strep. So again, pretty significant overgrowth pattern. We'll talk about the possible relevance of that as well. In the autoimmune triggers, we can see that Klebsiella was flagged as high. That is a marker that generally is associated with potential for inflammation, so that may be relevant overall. Under the fungi yeast section, we didn't see anything detected. Also no enteric viruses. As far as the parasite section, we also didn't see any protozoa, and also the worm section was negative. And if we look at the intestinal health section, definitely some significant imbalances occurring here with low elastase, high secretory IgA, and elevated calprotectin. So we're going to kind of parse through this particular case and provide a lot of background information, especially based on some of the emerging research that can help give us some really key insights into how this all might tie together and basically help provide clinicians with a more comprehensive picture of what's going on for patients like this. So if we look at the summary here, just to kind of bring it back around, we saw that there was a detected level of high C. diff based on the toxin genes. Overall, there was, quote, kind of a dysbiosis pattern. We'll get into the details there, but essentially a mix of high and low bacteria, especially in the normal bacteria and opportunistic sections. High calprotectin was noted, indicating inflammation in the gut, along with high secretory IgA, and then again, low elastase, which is an indicator for low pancreatic enzymes. So really the key questions for a lot of clinicians when they see results like this is, of course, is the C. diff significant for this individual patient or client? Also, are there other imbalances that may be related, such as dysbiosis, digestive markers, immune responses, et cetera? We do see that those are present for this patient. Again, the question is how may they be related to the presence and especially the high level of C. diff in this patient? And of course, overall, what is this picture telling us? So that's what we're really hoping to help provide answers for you today based on what we know from research and what we often see clinically with GI map. So we can get a lot of clinical insights into C. diff based, of course, on what we already know about C. diff and what's been established for a number of years in terms of just the characteristics of C. diff, the typical clinical effects when it is causing a significant infection. Certainly we know what the common symptoms are, et cetera. So a lot of that information is something that's been around for a while, but we're getting a lot of new insights from recent research, as I mentioned, which we'll talk about. And then once again, the common C. diff related patterns that we often see with C. diff, and we're going to talk about that again here as well. So as far as the basic information on all of our markers, including C. diff, I would direct you to our website, and in particular under the clinicians tab. Here you can see in the drop-down menu that one of the subsections is highlighted, and that's the Learning Resource Library. Highly encourage clinicians to check out our resources in that section. If you're new to GI map and are also just not very familiar with C. diff, I would certainly direct you especially to the interpretive guide. That's one of the resources that you'll find on that resource library page. Within the interpretive guide, we do have a section explaining some of the basics of what's known about C. diff. So starting with a few things here, I just want to kind of focus on. You can see under the epidemiological information, the statement is 2 to 10% of the population are carriers of C. diff, and most are asymptomatic. And that's a really important point to note because obviously a large proportion of people that have detectable C. diff do not have any of the classic symptoms of C. diff infection. So in most cases, certainly in conventional medicine, that's not really considered something that needs to be focused on from a therapeutic standpoint. But there is a question from newer research coming out as to whether that's still potentially telling us something and whether or not there may still be some subclinical type impacts from an overgrowth of C. diff. So with regard to asymptomatic infections, this is some additional information. And you can see the reference at the top here. So this is a general review paper on C. diff infection. And in this paper, they say in hospitals, the prevalence tends to range from between 7 and 18% in different studies. And that depends on a number of factors. So they say here it depends on the length of the stay in the hospital, exposure to antibiotics, as well as to, of course, C. diff itself, the presence of C. diff spores essentially in the hospital environment, as well as the presence of underlying disease. And then there's also been a link in research, which we'll talk more about later, to acid suppressive medication, especially proton pump inhibitors that may increase the risk for C. diff infection. They go on to say asymptomatic colonization out in the community versus hospital-associated infections is lower, of course, than in the healthcare setting. It's typically in the range of 2 to 4%. Now, with GI map, based on our internal data, we tend to see our numbers definitely within that range typically. I think the latest data that I've seen is that typically our ranges are in the 3 to 4%. So right in line in terms of what is typically seen in these other studies in the population in general. Now, this is another interesting thing to note, especially for those of you that work with children. Says several studies report higher carriage rates up to 25 to 30% in children in the first year, and that the rate drops down to around 15% once they're older than a year old, in that sort of range from 1 to 8 years. And they've explained this as this, based on what they say here, this variation is probably associated with the unstable intestinal microbiota in the first 2 to 3 years of life, which may enable C. diff to remain established in the intestinal tract. So that's really an important thing to note, that it may not necessarily be a big issue for some young patients if C. diff is detected, because the microbiome generally is still maturing for most children up until around the age of 3 or so. So many of those bacteria that we know that normally help protect against various infections, including C. diff, may not really have established their full levels yet in those young patients. And that, of course, in a relatively healthy scenario with a proper diet, then that's likely to become established. And then that may result in reduced or even elimination of C. diff carriage. So just a few interesting things here to note. And again, asymptomatic carriage is quite widespread, and that tends to be clinically what we see most often in our clinical consults relating to GI map. Just as a reminder, this is information directly from the CDC on the classic symptoms of C. diff, just for comparison. So essentially, C. diff infection, the symptoms often tend to happen within a few days after taking certain antibiotics. So it's definitely considered an antibiotic-related infection, and that's one of the key risk factors for C. diff infection. Typically, the symptoms of really significant infection would be severe diarrhea, fever, stomach tenderness or pain, loss of appetite, and nausea. And again, most of the asymptomatic carriers, of course, by definition, don't have these classic symptoms. So back to our case example here, and just sort of reminding you of the situation here. For this particular patient, of course, one of the key symptoms was chronic constipation. So really the opposite of what's typically associated with an outright C. diff infection, plus none of the other symptoms, as you can see here, that are typically associated were reported by this patient. So definitely appears to be more of an asymptomatic carrier type scenario. So the question is the C. diff really having an impact? And also, what may the presence of C. diff be telling us? One of the recommendations or suggestions in our interpretive guide under the therapeutic approaches and considerations section, in terms of really assessing the patient and the potential impact of C. diff when it's positive, is to really look for the patient's calprotectin and also the secretory IgA levels to get an idea of the immune response. Now, of course, that has to be interpreted within the context of the rest of the test, and there may be other factors that could also help explain elevations there. So just because C. diff is there and calprotectin is elevated doesn't necessarily mean that C. diff is causing the inflammation. There may be other factors. So that's really where careful assessment comes into play. You can see here for this particular patient that we've been talking about, the secretory IgA was elevated just a little bit beyond the high end of the range, but calprotectin was significantly elevated. So certainly that's suggestive that C. diff, even though it's not causing classic symptoms, might still be having an impact. So let's actually look a little bit more in detail at what's known about the impact C. diff when it's causing significant issues. So this information is from an article, review article. You can see the reference at the top here. Title is Clostridium difficile colitis, Pathogenesis and Host Defense. So in the virulence factor section, they say C. diff produces toxins that primarily target the intestinal epithelial cells following toxin endocytosis and activation in the cytosol, i.e. inside the cells, which are the epithelial cells. Those cells can undergo necrosis, so it's a type of cell death, which leads to, of course, loss of intestinal membrane integrity, and then exposure, a greater exposure to the normal intestinal microorganisms, which can then result in activation of an inflammatory response. So that's kind of a quick summary of what's known about the pathogenic process and the role of these toxins in disrupting the barrier and then allowing those organisms to get through and then provoke an immune response. They go on to say a few more details about the particular toxins that we have as markers on our test, which again are the toxin A and B. It says the major toxins that are encoded by the pathogenicity locus. That's the part of the C. diff genome that encodes these and other toxins and other virulence factors. So essentially the main ones are called TCDA, which is also known as toxin A, and TCDB, also known as toxin B, which are two large secreted proteins. So these are really the two main toxins that C. diff produces. They go on to say that these toxins disrupt the cytoskeleton and then lead to dissociation of tight junctions. Of course, those are the factors that help prevent leaky gut, but those can be disrupted by these toxins between the colonic epithelial cells, and that's another factor that leads to the loss of the epithelial integrity. So essentially these toxins do a couple things, but they increase leaky gut essentially, and then depending on how severe the scenario is, they can also cause cell death. And all of that together can cause pretty significant damage to the intestinal lining, which then causes inflammation. Just a visual from that article here for those of you who are more visually oriented. So this is basically showing you a schematic of C. diff in the colon, releasing these toxins, toxin A and B, that are basically disrupting the barrier here. So basically once that happens and that causes epithelial damage, you can see they've shown that microbes tend to get through then into the underlying area, which is the lamina propria, which is of course where all of the immune cells are. So once those microbes are detected in this area where they're not supposed to be, then that can essentially stimulate a pretty significant immune response, and that's illustrated here. So that's essentially what's happening at the sort of core of the pathogenic process. And just a note here, back to our interpretive guide under the C. diff section, you can see here it says that the GI map tests only for the genes for toxin A and B, which are carried by C. diff. The GI map does not measure the toxins directly for C. diff or any other microbes. So it's the GI map test is focused on DNA, which of course is reflecting the gene content, but is not reflecting whether or not those toxins are actually being expressed. So that's really where symptom assessment is really important, and then also potentially getting some clues from the test itself, as we talked about, with calprotectin and other markers. I just want to talk a little bit more about that concept here, that typically, even though pathogens, of course, by definition, have the ability or the potential to cause infections and pathology, they don't always do so, and it really depends a lot on whether or not and when they express their virulence factors and at what level. So there's a lot of essentially sort of regulatory effects that happen to control toxin synthesis for various pathogens. So what they say here is the symptoms of C. diff infection are essentially caused by the production of these two toxins, toxin A and B. Moreover, for severe forms of disease, the spectrum of disease caused by C. diff has also been correlated to the levels of toxins that are produced during host infection. This observation strengthened the idea that the regulation of toxin synthesis is an important part of C. diff pathogenesis. So really, it's not so much just the presence of the toxins. That's telling you that the potentially pathogenic strains are present. And then the levels are telling you that that strain is growing at a certain level. If you only see sort of a trace amount, then that's indicating that the pathogen is present, obviously at a lower level, and is unlikely to produce large amounts of toxins. But at higher levels, there's a greater potential. So really, in addition to overall expression of the toxins, the overall levels of the pathogen are really important in terms of whether or not that can potentially result in significant issues. So all of this kind of really leads to this concept that there are many factors that can influence the colonization of pathogens, their growth, and the virulence, and in particular, that's true for C. diff. And based on a lot of new research, we have some great insights into those factors, and we'll talk about some of those today. So essentially, those factors can be categorized in several areas that are very relevant for functional and integrated clinicians, because these are highly potentially actionable areas, particularly diet, also digestion factors, and then the microbiome. So these are really three key areas that are known to help influence the growth, colonization, and virulence of C. diff. And that's really kind of part of this general concept that's emerging from new research, where sort of the old model was just identifying whether pathogens, opportunists were present, and then if so, and if that correlated with significant symptoms, then oftentimes that would warrant direct treatment. But we're learning through all this new research, particularly in terms of the microbiome, and understanding the interactions of the microbiome with the gut environment, and then how those factors then interact with pathogens, again, that there can be a lot of influence there in determining whether or not pathogens are successful in mounting an infection. So that's really the role of GI-MAP, because we, as a comprehensive stool test, we provide a number of markers that give you insights into both sides of that equation, the microbial ecosystem, as well as GI physiology. And this is just sort of the detailed categories of markers. And of course, most clinicians initially tend to be more focused on presence of pathogens and opportunists, but it's really important to take the rest of the picture into account. And as we go through the rest of the webinar, hopefully you'll understand some additional ways in which these markers can inform what's going on with the various pathogens and opportunists. A lot of that is reflected in our dysbiosis patterns that we've recognized with GI-MAP over the years. And this is a set of patterns that we commonly see. Sometimes patients have just one pattern. Sometimes they may have a combination of more than one or all three. And these types of patterns are also something that's seen quite a bit in research. So there's a lot of research evidence supporting these different types of dysbiosis coming into play when it comes to a variety of infections as well as chronic diseases. So let's start off and just really briefly talk about inflammatory dysbiosis. So that's the one that's most directly relevant to pathogens. And of course, that's one of the things that you want to assess for if you do see pathogens like C. diff. So whether there's really any evidence of inflammation, because that's often a common consequence of the action of pathogens. So in this case, for this patient, you can see there's a high level of C. diff, both toxin A and B, and also the calprotectin was detected at a high level. So that's confirming that inflammation is an issue here. Doesn't necessarily confirm directly that that's caused by C. diff, but it is evidence indicating that that could be a cause. So that's really all we need to talk about there as far as inflammatory dysbiosis. That one's fairly straightforward as far as the connection between pathogens and inflammation in the gut. And sometimes you may see a related inflammatory type pattern in the microbiome. So there are quite a few markers on GI-MAP that have the potential to also be inflammatory. In many cases, when pathogens cause inflammation, that can cause a broader inflammatory dysbiosis. So now we're going to talk about this insufficiency pattern, which is essentially a lack of beneficial bacteria. This is a pattern that we commonly see when C. diff is detected. So I'll show you a number of examples here just to kind of emphasize how common it is. So this is a patient that had detectable C. diff toxins. One was high, one was moderate. This patient had a very broad deficiency pattern in the microbiome. So this would be a fairly significant pattern where you can see quite a few of these markers in the normal bacterial flora section are quite low. So that's based on everything we know, that's likely very relevant to why C. diff is there even in the first place. That can be a predisposing factor to higher levels of C. diff. So again, you can see there's quite a few here. Just note that in this section that we do have markers such as the two here for Akkermansia and Faecalibacterium that both say less than DL. So just keep in mind that less than DL does mean that those markers are quite low. In fact, less than DL means less than detection limit. So definitely include anything in this section that says less than DL in the picture of potential deficiency. This is an example from another patient, also had high levels for both toxins. You can see in this case, there's a little bit of a mixed pattern, not as strong as the last one. In fact, there are a couple that are flagged as high, but at the same time, we can see that 2 of them are flagged as low. So you may see this type of scenario too, where it's not as broad based in terms of the deficiency, but yet there can still be some pretty significant and important groups or species that are deficient in these cases. All right, so this is yet another example with quite high levels, E6 and E5 levels for toxin A and toxin B. And then if you look at the normal bacteria section here, again, you can see several groups that are flagged as low. So there's the Clostridia in this case, once again, Akkermansia, and then also Faecalibacterium. This is yet one more case, just to kind of illustrate how common this is when we see C. diff. So this is quite a high level of C. diff in this patient, both toxins at the E6 level. And then once again, we can see Akkermansia, Faecalibacterium are low. The Bacteroidetes phylum here is also low. So again, pretty significant deficiency, but in the context of also elevations in some of the other markers. So sometimes you will see a mixed pattern, but still important to take into account the ones that are deficient. So I kind of want to add some context around this. So we'll talk more about the potential importance of the deficiency in relation to C. diff. But more directly, we certainly know that antibiotics tend to be a major risk factor for C. diff infection. Of course, by definition, antibiotics can reduce not only the target organism, but also more broadly, a lot of bacteria in the microbiome. And really the impact of antibiotics is going to be dependent on the individual antibiotic and their spectrum of activity, whether they're broad spectrum or narrow, also the dose and length of time and factors like that. So a number of antibiotics have been shown to predispose to C. diff infection. So that's another piece of evidence indicating that when you reduce beneficial bacteria, that potentially can predispose. So the question is why? We also know that FMT, or fecal microbiota transplant therapy, also is very successful in treating C. diff infections. And of course, by definition, that's essentially restoring a relatively healthy microbiome compared to the microbiome that the patient had. So lots of research has been done into that as well. And it's pretty clear that FMT therapy likely works by helping to restore beneficial bacteria. So just based on these two observations, we can tell that likely normal commensal beneficial bacteria are quite important for helping to prevent C. diff infection. So really the question is, what is the role here? So this gets into the general concept of what's called colonization resistance. So this is a review paper from a few years ago titled Control of Pathogens and Pathobionts, which are essentially opportunistic pathogens, by the gut microbiota. And they say a major function of the microbiota is protection against colonization by pathogens and overgrowth of indigenous pathobionts that can result from the disruption of a healthy microbial community. So that's really kind of a central feature of lots of different infections beyond just C. diff, that when the microbiome is disrupted, and especially in a way that reduces key beneficial bacteria, that can provide an opening for pathogens to then colonize and potentially infect the gut. So they go on to say that pathogens in turn have evolved strategies to escape from commensal-mediated colonization resistance. Thus, the interplay between the commensals and pathogens, or the indigenous opportunist, is critical for controlling infection and disease. So this is really a very important point to understand, and why we want to look at the overall microbiome picture, because that can give us a lot of information in understanding why those pathogens are there, potentially even in the first place, but also suggest potential supportive or therapeutic options in order to help to inhibit those pathogens by increasing the beneficial bacteria. This is a schematic from that article, illustrating kind of visually what we just talked about. So on the left is a healthy microbial community. They're showing the pathogen coming into the system, in this case C. diff, or other pathogens, and that basically the normal microbes shown here kind of at the center are producing a number of different factors that can inhibit them, and that's shown by these lines with the essentially blocking the pathogens in various ways, versus in a disrupted community with antibiotics that result in depleted microbiota. That can lead to overgrowth of not only opportunists, but of course, less competition for the pathogens that are coming in, and then they have easier access to the epithelium where they can start mounting an infection. So that's kind of a classic scenario that's seen in a lot of different gut infections, not only C. diff. So this particular article is titled C. diff, colitis, pathogenesis, and host defense. So they talk about one of the potential mechanisms here. And so far to date, there are a number of potential mechanisms that have been shown in terms of how the normal microbiome protects against pathogens like C. diff. So they say here the intact microbiota converts primary bile acids into secondary bile acids, several derivatives of which can inhibit the growth of C. diff. So essentially bile acids that of course are coming through from the gallbladder into the GI tract. Typically, especially when the residual bile acids, most of course are reabsorbed in a healthy gut in the ileum, but a small percentage, even in a healthy person, does get into the colon. And even within the small intestine itself, these still can be relevant for C. diff. But essentially a healthy microbiome helps convert those primary bile acids into secondary bile acids. So it basically modifies them in a way into a different form, and that secondary bile acid set of products then can inhibit C. diff. And they go on to say the antibiotic-mediated disruption of the microbiota depletes the primary bile acid converters, which enables C. diff sporulation and growth. So that's thought to be one of the major ways in which antibiotics can predispose to C. diff infection. And they just show that visually here in this figure, where you can see at the top it says undisturbed or healthy microbiota. And then I highlighted on the left, the commensal bacteria tend to convert these primary bile acids into the secondary bile acids, and they show that inhibiting the growth of C. diff. Whereas in the antibiotic disrupted microbiome, there's either no longer sufficient levels of those beneficial bacteria, or in some cases, the antibiotics may have completely eliminated them, so that it's not possible to convert the primary bile acids into sufficient amounts of the secondary bile acids. So there's sort of a lack of an inhibition then on C. diff, which would then, of course, result in greater growth of that pathogen. This is an article kind of sort of addressing a general feature that's been shown in a number of different pathogenic infections. The title here is a dietary fiber deprived gut microbiota degrades the colonic mucus layer and then enhances pathogen susceptibility. And then they have a nice summary here, visual summary of sort of the conclusions from that research, which is, once again on the left, is a healthy gut based on a fiber-rich diet that's promoting adequate amounts of normal microbes. And the action of those normal microbes through producing things like short-chain fatty acids helps to generally promote a healthy intestinal lining, especially the mucus layer, that helps keep the microbes away from the epithelium. So it makes it less likely that they're going to be able to infect. But then you can see on the right here that just a fiber-free diet, even in the absence of antibiotics, can have a pretty detrimental effect on the gut barrier. So they're showing that essentially with less fiber, you have less normal microbes, and then pathogens have an easier time of competing in the gut. Plus, then you have less stimulation from those normal microbes of a healthy intestinal lining. So that can all kind of compound in making it more likely that these pathogens can cause a significant infection. So I just want to point out here on GMAP, and again, this is a screenshot from the case that we opened with. You can see here in the normal bacteria section, I'm highlighting the marker for Akkermansia mucinifila, which in this patient's case was less than detection limit. This is widely regarded as a potential marker reflecting the health of the mucus layer. And that's primarily because Akkermansia depends completely on mucus as its main food source, as its main source of energy. Lots of studies have shown that conditions that are featured by lack of mucus, such as ulcerative colitis, also tend to have lack of Akkermansia as well as one of the key features. So that's a general indirect marker for the potential health of the mucus layer. So this is indicating that that could be one factor that's happening for this patient. There's less protection. This is a really important study that was published a few years ago. It was in a mouse model, so this is not a human study. And a lot of this research, of course, is more in the basic research realm versus clinical. But if you look at the title here, it says microbiota accessible carbohydrates suppress C. diff infection in a mouse model. So their term essentially for fiber is a little bit more technical. They call it microbiota accessible carbohydrates, or MACs. So I just want to look at some of the information that they have here in their summary. And they say, although diet is among the most powerful available tools for affecting the health of humans and their relationship with their microbiota, investigation into the effects of diet on CDI, which is Clostridium difficile infection, has been limited. Here we show in mice that the consumption of MACs, or fiber, found in dietary plant polysaccharides has a significant effect on C. diff infection. Specifically, using a model of antibiotic-induced C. diff infection, we demonstrate that the MAC-deficient diets perpetuate C. diff infection. We show that C. diff burdens are suppressed through the addition of either a diet containing a complex mixture of MACs, i.e. fibers, or a simplified diet containing just inulin. So let me skip back there. Containing inulin as the sole fiber source. So again, they showed here that essentially diet deficient in fiber can promote C. diff infection, and then adding fiber can help protect against C. diff infection. So they go on to say in their conclusion that our findings, when placed in the context of known fiber deficiencies in the typical human Western diet, provide rationale for pursuing a fiber-centric dietary strategy as an alternate line of investigation for mitigating C. diff infection. So it's a really active area of research in terms of how do we promote the normal beneficial microbiome functions that help protect against C. diff infection. Just a number of other studies are really coming out to support this general concept. This is another example. This one is titled Butyrate Protects Mice from Clostridium difficile-Induced Colitis. And then they mention a particular molecular mechanism that was shown in this study through HIF1 dependent mechanism. So this is their summary, which says that their findings were that butyrate is able to protect the intestinal epithelium from the damage caused by C. diff toxins by stabling this particular factor, HIF1, and then increasing tight junctions, which reduces intestinal permeability, thus inhibiting inflammation and bacterial translocation. So it's essentially not necessarily directly counteracting the pathogen, at least in this particular case. They showed that it was mitigating some of the damage that C. diff was causing, and again, having a beneficial effect in this case. Yet another example, in this case, Faecalibacterium prausnitzii plus prebiotic were shown to help protect intestinal health in a mouse model of antibiotic and C. diff exposure. So just wanted to highlight that because, again, we're coming back to this particular case that we've been talking about. You can also see that Faecalibacterium prausnitzii was deficient. We know that that's a key butyrate producing bacterium. We just saw evidence that butyrate can be protective. And also that particular study showed that administering Faecalibacterium in a mouse model also can be protective, especially when it's provided along with fiber, a prebiotic. Just a quick another example of yet another commensal microbe that has been shown to be protective. In this case, Bacteroides fragilis, which is also on GI-MAP. That species was shown to prevent C. diff infection in a mouse model by restoring the gut barrier and also general microbiome regulation. So again, a lot of accumulating evidence, not only that in general beneficial microbes can protect against C. diff and other infections, but also getting into the details of specifically which species, which microbes, and also how they are potentially protecting against infections. So really that kind of illustrates just how important this lack of beneficial bacteria or insufficiency dysbiosis can be for patients who exhibit presence of pathogens and especially signs of infection. And again, it's really important to assess insufficiency dysbiosis when you do see those scenarios. The third common dysbiosis pattern that we typically observe in many patients with GI-MAP is the digestive dysfunction or overgrowth dysbiosis type pattern. This is something we also often see with C. diff and also may be giving us some information into the gut environment that might be contributing to the presence and overgrowth of C. diff. Again, you can see for the patient we've been focusing on, the patient case, that elastase was low, and that's direct evidence that at least one aspect of digestion is insufficient for this patient. That's well below the 200 cutoff, indicating that pancreatic function is insufficient. Again, this is something that we often do see with C. diff infection, or the presence of C. diff on GI-MAP, and also more broadly with dysbiosis in general and presence of pathogens. We also tend to see an overgrowth pattern, and you'll typically see that most prominently in the opportunistic bacteria section. So again, this is the opportunistic section for the patient case that we've been focusing on. You can see a very extensive amount of overgrowth with these different markers. So a pretty significant pattern here of overgrowth, and it's something we often do see in patients that have evidence, direct or indirect, of potential poor digestion. Just want to give you another example here. This is a patient that had also detected C. diff. In this case, only one toxin was elevated. And you can see here that the elastase was quite low, and also the secretory AG was quite low in this other patient. So again, this is a pattern that we commonly see. So just keep in mind that the low elastase, is of course reflecting one aspect of digestion, and we do see that often. But also secretory IgA can be seen as a general marker for lack of beneficial bacteria as well. So we often do see that deficient in many patients that have a lack of beneficial bacteria. And it can be often, again, taken as a marker suggesting lack of beneficial bacteria, mostly because bacteria, normal bacteria in the gut, are largely responsible for promoting normal levels of secretory IgA. So now I want to kind of broaden the picture here a bit more about what's known in research that can be related to negative aspects of digestion and how that can relate to C. diff. So there's a number of studies that have been conducted over the years that have shown a link between patients who are using proton pump inhibitors and an increased risk for C. diff infection, as well as other infections. And this is just one example. And this is a review paper, as you can see by the title, Risk of Long-Term PPI Use, a Critical Review. They say this increase in PPI usage over the past 2 decades has called into question the long-term effects of these medications. The association between PPI use and infection, particularly C. diff infection, and also pneumonia, has been the subject of several studies. It's proposed that the alteration in the gastrointestinal microflora by proton pump inhibitors produces an environment conducive to the development of these types of infections. So they're implying that it may be sort of the dysbiosis or overgrowth that's often a result of low stomach acid, and we do often see that, once again, as part of that digestive dysfunction overgrowth type pattern. But there may be some effects directly of inhibiting acid. That's addressed in a number of other studies, such as this one, that talks about the impact of pH on C. diff sporulation and physiology. And then in that article, this is their abstract, And I highlighted a couple areas here. So they say in the first highlighted area that we observed pH-dependent differences in sporulation rate, spore morphology, and viability. Sporulation frequency was lowest under acidic conditions. So again, when acid levels are healthy, then that suggests that the frequency of sporulation is going to be generally lower. So when acid is insufficient, likely it's going to be higher. That's been shown in other studies as well. So they say at the end here, the data demonstrate that pH is an important parameter that affects C. diff physiology and may reflect or may reveal relevant insights into the growth and dissemination of this pathogen. And then a similar research article concluded essentially kind of the same thing. This was titled Germinant Synergy Facilitates C. diff Spore Germination Under Physiological Conditions. So they say in their summary that we propose that the acidic conditions within the GI tract regulate C. diff spore germination and can provide a biological explanation for why patients taking PPIs are associated with an increased risk for developing a CDI or C. diff infection. So once again, potential links between hypochlorhydria and an increased burden of C. diff. So yet another aspect of digestion that may contribute to this issue. And related to that potentially, but mostly shown in terms of dietary factors, is the potential role of excess amino acids in the gut. And of course, that can result from high protein intake and or not digesting the protein very well. So the title here is C. diff uses amino acids associated with gut microbial dysbiosis in a subset of patients with diarrhea. They go on to say, protein-based dietary interventions are partially protective in animal models of C. diff infection, suggesting that a proline-deficient diet could be a simple, actionable therapy for providing protection to at-risk individuals. So just kind of a side note, we're going to look at a few other studies that provide kind of additional evidence supporting this idea that excess amino acids getting into the colon, again, either from a higher protein intake or from not digesting well, may be part of the picture in promoting the growth of C. diff. There's a number of research studies that really focus in on that particular amino acid that's mentioned here, which is proline, which tends to be elevated in certain types of foods, especially animal foods containing connective tissue. So for example, collagen supplements, bone broth, etc., that may have higher levels of proline. There's some evidence now to indicate that especially sources that are higher in proline may be especially effective in promoting the growth of C. diff. This study basically was in an animal model, but basically they looked at kind of a high-fat, high-protein Atkins type diet in this animal model and shown that it basically promoted C. diff infection. And they also showed that a high-carb diet, i.e. a higher fiber diet, protected. So there's, again, growing evidence here that this balance in fiber versus protein, for example, can play a role in influencing the levels of C. diff in the gut. This was an interesting study. It was just recently published in January, where they looked at a number of different factors that were associated with C. diff infection. So the title is Association of Household Pets, Common Dietary Factors, and Lifestyle Factors with C. diff infection. So one of their hypotheses was that household pets may help to provide a richer microbiome just by increasing the microbes in the environment, and that may be protective against C. diff. But they say in their conclusions that they found that living with a cat or a dog was not associated with negative CDI testing, i.e. protection, but they did note a few other things. It says several novel risk factors for C. diff infection have been identified, including high meat intake and then also cat allergies, interestingly. But again, I really wanted to emphasize the increased protein intake because that's a common theme that we've been seeing in a lot of the research coming out on C. diff, as well as other potential gut pathogens. So high protein diets certainly have benefits under certain scenarios, but certainly when it comes to these types of infections, there may be a case for at least temporarily, reducing protein intake so as not to promote the growth of certain pathogens. So this is yet another article on these lines. The title here is C Diff exploits toxin-mediated inflammation to alter the host nutritional landscape and exclude competitors from the gut microbiota. And again, this is also very recent from January 2021. In their summary, they say, host gene expression signatures suggest that degradation of collagen and other components of the extracellular matrix by these enzymes, matrix metalloproteinases from C. diff, is a major source of peptides and amino acids that supports C. diff growth in vivo. So yet another line of evidence suggesting that, once again, amino acids, sources of protein, may be particularly significant. But they mentioned specifically, of course, collagen. That's a major factor in the extracellular matrix and a key source of proline. So just anecdotally, in a number of the cases that I've reviewed with GI map, when I mentioned this potential connection between proline and C. diff, and that proline is in high concentration in certain foods such as collagen, a number of clinicians have mentioned that their patients had been taking, for example, collagen supplements or consuming bone broth, et cetera. So it is something that potentially, again, this is just really early research. It's not really definitive yet, but there are many different studies that are starting to show that higher protein, especially the higher proline containing foods, may promote the growth of C. diff. So it may be wise to avoid those, at least during the treatment period. So that brings me back to our case here. And really just kind of looking back at all the different features that we saw for this patient. Now that we've gone through some of this research and looked at not only just the presence of C. diff, but what else is going on here in terms of the lack of beneficial bacteria in this patient, the overgrowth, digestive dysfunction type pattern, the lack of elastase, et cetera, plus of course the calprotectin, which suggests there may be more of a significant impact from C. diff in this case. All of those are really in line with this emerging research suggesting once again that the gut environment, including the microbiome, can have a really big impact on influencing the presence, the colonization, and the growth of C. diff. So those may offer additional avenues for clinicians in terms of how you approach addressing the C. diff that's present. So again, this is just kind of a quick last run through that report for this patient. So now that you have all this background information, when you look at these results, you should be able to really get a lot more out of what you're seeing here. So high levels of C. diff, once again, not a massive deficiency pattern in the normal bacteria section, but we did see Akkermansia was low, again suggesting possible reduction in mucus. Faecalibacterium presenitzii was low, and again, that can lead to potentially lower butyrate levels, and there was the study that showed that it does help protect against C. diff infection. Of course, it's deficient here. Again, an overgrowth pattern, typically consistent with reduced digestion. Klebsiella was detected. That can be a contributor to inflammation, so it may be one of the contributors to the high calprotectin. But also these inflammatory microbes often, as I mentioned, can become elevated once there's a pathogenic infection. So pathogens can come in, cause inflammation, and then that inflammatory scenario can then promote the growth of other inflammatory type microbes. That's often seen in various types of infections. So this, no way to tell for sure because there isn't a before and after test before C. diff infection. So we don't know if the Klebsiella was there prior to C. diff, but it's possible this might be a result of the C. diff infection. And again, looking at the intestinal health section, elastase was low, so that's confirming reduced digestion, which could potentially lead to increased undigested protein or insufficiently digested protein getting into the colon. And then again, that can be another factor promoting the growth of C. diff, and then essentially resulting in these immune responses from the action of C. diff. All right, so hopefully that kind of illustrates this general concept that's emerging from research, that we really don't want to just focus exclusively on pathogens, but we do want to take into account the rest of the picture in the gut, because that can really go a long way towards helping to inform us in terms of the conditions that may be favorable to the gut or to the pathogen, and then what areas that we may want to focus on in terms of therapeutic approaches. So once again, it's important to kind of understand these different patterns on GMAP and how to recognize them, because that can go a long way towards helping to give you insights into these imbalances. So once again, it's all about learning how to not only just understand the individual markers, but then how to connect them. So it's a lot of this sort of more advanced interpretation has to do with how do you connect the dots based on all this emerging information. With that, I want to wrap up the webinar and thank you for attending. And it looks like we may have some time here for some questions.
Yes, we do have time for a couple of questions. The first one that we have is, can inflammatory dysbiosis also be from bacteria overgrowth in the absence of pathogens?
Yes, certainly that's been shown in a number of studies. So of course the pathogens on GMAP are essentially on page 1. And then throughout the rest of the test, there are other what are generally referred to as opportunistic pathogens or opportunistic microbes that typically can be normal residents of the microbiome. So you'll actually see a couple of those inflammatory microbes in the normal bacteria section. So the two prominent ones would be the one called Escherichia, and then the second one would be called Enterobacter. And both of those are part of they're in what's called the Proteobacteria phylum. And then within that phylum, there really is a group that are largely associated with inflammation. So they're known as sort of the inflammatory LPS producers, for example. So the Escherichia and also the Enterobacter are part of that group, and they do produce LPS. They do have beneficial functions, so really they're not considered much of a concern unless they're significantly elevated. Really, most of the microbes that are opportunistic that can be inflammatory would be in the opportunistic section. In that first section of the overgrowth dysbiotic bacteria, primarily that would be Morganella and also Pseudomonas. Particularly with Pseudomonas, there's recent research showing that that can cause significant gut inflammation, usually more in the small intestine. In that case, that might contribute to food sensitivities. And then especially in the autoimmune triggers section, that's really where the largest concentration of potentially inflammatory microbes is located on GMAP. So organisms like Citrobacter, Klebsiella, Mycobacterium, and Proteus in particular have all been shown to be not only potentially inflammatory and also associated with certain autoimmune conditions, but also have been generally associated with inflammation. So sometimes when you see those elevated, even in the absence of any pathogens, you can still see that calprotectin is elevated in some of those cases.
Great, thank you. That is all the questions we have time for today. So thank you, Dr. Fabian, for the great presentation you just gave. Questions asked during the webinar will be answered by Dr. Fabian after the presentation. So thank you everyone for attending today.
Thanks so much.